Great. Thanks, Carrie. Appreciate the opportunity to be here to talk to all of you about work we've been doing recently on new methods to identify very comprehensively structural variation, particularly in cancer genomes. There we go. So as all of you know, cancer is a somatic genetic disease, often manifest as seen in this sky-carry type of a cancer cell line of structural variation involving translocations, insertions, deletions, insertions, and aneuploidies. As you heard from Faisal earlier this afternoon, identifying many of these structural variants is critical in the prognosis and treatment selection for cancer patients in a number of different cases. The difficulty is being able to identify all these comprehensively and to do so in a method that allows it to be readily used in the clinic. And what I want to do is talk about some new methodology that allows us to, I think, bring a new approach to developing a comprehensive view of the structural variation in cancers. And so the idea is to build a cancer genome from scratch, that is without using a reference genome. But the challenge using technology such as next-gen sequencing is that's very limited due to the number of repetitive sequences within the human genome, and particularly within cancer genomes. So the approach we've taken is to apply two complementary tools, or in the first case I'm going to talk about today, just one new tool, and that is optical mapping using a new technology developed by Bio Nano Genomics. And that's coupled with, in many cases, with whole genome sequencing. Since everybody knows what whole genome sequencing is, I'm going to focus for a few minutes on explaining optical mapping. The methodology is based on actually directly visualizing ultra-long DNA molecules in their native state. And then these consensus assemblies are used to accurately call structural variation between samples and the human reference genome, or if you are dealing with a sample where you can get the normal genome from that individual, you can look at the variation between the samples and the normal genome. This resolution is 1,000-fold higher than chromosome banding from karyotype analysis, and it's suitable for very complex and balanced rearrangements. So this is the basis of the technology. It's nanofluidics, and uses some technology actually developed at Princeton a number of years ago, in which a large DNA molecule is run through nanochannels that looks like this device in the center here. The initial area of this microfluidic device are a set of pegs that unwind very large DNA and then introduce it into a series of nanochannels showed here, where the nanochannels are no wider than the width of the DNA. So the DNA that enters these nanochannels have to assume a strictly linear form. And so this is the instrument on which it's done. And the trick in all this is isolating high molecular weight DNA, which we can now do from liquid cells, liquid cultures, or from solid tissue. Prior to running it on this device, we labeled the DNA with an enzyme that transfers a fluorescent dye to any site at which a particular 6-base sequence resides. So this basically barcodes every DNA, and then that DNA is applied to the chip. This is what the DNA looks like as it's running on the chip. So the DNA is electrophoresed into the chip. It goes through these pegs at the top that then unwind it. and send it through a series of decreasingly smaller nanochannels until we get to the point that each of these is a single molecule stretched out in a linear array along the channel. Those molecules are photographed during the process of transiting the nanochannels. And this is the data that is obtained from those images. As you can see, there are streaks of DNA. Each of these blue streaks is a DNA molecule, and you can see on the blue streaks the fluorescent dye that marks the position of that particular 6-base sequence that targeted the transfer of the fluorescence. So what do we do with that? Well, we take those images, digitize them, and then with those digitized images, we look for overlap. And from that overlap, we just build a consensus map. And that consensus map, then we can use to compare either to the normal genome of an individual or to a reference genome to identify insertions or deletions or any other structural variation. And this is the assembly that we would get from that approach. So each one of these orange lines at the bottom of the figure is a DNA molecule where the black ticks are the presence of that fluorescent marker. And you can see these molecules can be aligned to assemble into a consensus map that in this case could be compared to the HG19 consensus reference genome to show that these molecules derive from position 125 megabases on chromosome 3. So I want to apply show our application of this to two different types of cancer. The first one is acute myeloid leukemia, and the second is HPV positive head and neck cancers. So focusing first on the acute myeloid leukemia, as we heard from Faisal earlier today, AML first is a fairly lethal disease. There are approximately 20,000 cases per year in the States with about 10,000 deaths. And as Faisal indicated, you can group these AML cases on the basis of the variations and the driver mutations that are underlying the progression of the cancer. And in this study from 2016, showing 1,500 patients, the results of 1,500 patients, each one of these horizontal lines is one patient, and the vertical line are the genes or structural variations that are found in that particular patient. And using this sort of analysis, there are at least 13 different subtypes that one can identify that characterize different prognosis based in this case on the European nomenclature to either high, medium, or low, as you heard before. And many of these driver mutations or changes that are used for classification are either translocations or aneuploidies that are readily determined by looking at structural variation. So also as indicated before, the type of structural variation that is found in a particular tumor has very strong prognostic behavior. So if you're a patient and are unfortunate enough to have AML, but fortunate to have that AML driven by an inversion on chromosome 16, the likelihood of survival after 5 years is quite high. On the other hand, if your AML is driven by an inversion on chromosome 3, the survival rate after 5 years is very low. So understanding the nature of the underlying driver mutations is critical for both prognosis and subsequently as a consequence of determining the aggressiveness of the therapy that's used. So as part of a consortium, a multi-center consortium, we've examined 100 AML samples, primarily to identify the extent to which bio-nanotechnology by itself is sufficient to identify all of the normal structural variants that are identified by standard clinical karyotyping with FISH and um, uh, chromosomal microarrays. So this is the way that data are organized coming off a bionano device. This is a Circos plot with the chromosomes ordered clockwise, starting at chromosome 1, going around through the sex chromosomes X and Y at the end. Translocations are shown in the center, indicating the beginning and end of that translocation. Copy number is shown in the inner ring here, indicating, for example, that this has one copy of X and one copy of a Y. So this is a male, whether this is a female. And then other point mutations, other structural variations, including insertions, deletions, and duplications are shown in rings just below the banding pattern on the outer ring. So this was a case whose karyotype was defined by cytology as normal, that is 46XY. But when we did the bionano imaging, in fact, it revealed that this had a translocation between 5 and 11 that wasn't identified by standard karyotyping. This is a case in which we have a very complex phenotype with a very complex pattern seen both by standard karyotyping as well as by the bionano. And I'm going to delve into this case in a short while. So in all, we see in these across these hundreds of patients, on average, a number of almost 20 deletions per case, almost the same number of insertions, a number of translocation inversions and duplications as well. So the bionano reveals a large number of structural variants in these 100 cases. They clearly pick up with much higher resolution the standard translocations that are diagnostic for AML, for at least prognostic for AML cases, including the 3-5 translocation. And as you can see from this resolution, we can see within 10 kb the sites of the translocation breakpoints. And here's another case of a standard 821 translocation that fuses RUNX1 to RUNX-T1. This also readily picks up the inversions. This is the inversion on 3, again with the breakpoints of the inversion defined within 10 kb or so. And then the other common inversion on AML was also found in these cases. This is the inversion 16. This also readily picks up copy number changes, so we can define aneuploidies, a case where we have a +8 plus N of 3 of chromosome 8 in this case. Here's a loss of chromosome 7, and here is a gain of 8, 9, and 14, but also indicating a small duplication increase in ploidy of a small portion of the right of the Q arm of 11 that wasn't defined by the standard karyotyping. So in all, we had about 48 cases that were normal by karyotyping, 16 that were abnormal with altered copy number, 27 with translocations or inversions, and 9 that were complex. All of these variations that were identified, all of these identifying clinical observations observed by karyotyping were also observed by optical mapping with a concordance of 100%. But in addition, besides concordance between the bionano, in 14 cases, the bionano resolved variant structures that were ambiguous by standard karyotyping or other technologies, reveal new variants in some of the cases, or both. And in 3 of these cases, these new observations changed the ELN classification either from a high risk to an intermediate risk or vice versa. So the data that we obtained from BioNano was both concordant with what was sees everything that's seen by karyotyping, but adds additional new information, in many cases, those that would in fact impact on the prognosis and treatment selection for those patients. Here's a case in which the BioNano identified a missing variant that was not observed by karyotyping. So the karyotyping revealed the translocation between 821, and the BioNano revealed, in fact, the same translocation. But in addition, the karyotyping identified what appears to be a translocation between 4 and 5. And if we look at the karyotype, there's no obvious change in the karyotyping structure that would indicate a 4 or 5 translocation. But if we look at this a little bit more closely, we can see that there is an increase in the copy number of the arm at the site beyond the point of the translocation of chromosome 4, and a decrease in the copy number of chromosome 5, again at the boundary of the other end of the translocation, suggesting that this portion of the Q arm of 4 has been translocated to replace a portion, equivalent size portion of chromosome 5. And this is confirmed by chromosomal microarray showing 3x copies of this arm of chromosome 4 and a loss of part of chromosome 5. In addition, it actually showed that the chromosome 811, the 821 translocation was also associated with a small deletion, which we seen on the BioNano that we confirmed by microarray. Here's another case of mistaken identity of a particular structural variation. In this case, this is one I referred to earlier, that the standard karyotyping identified translocation between 8 and 9, X and 9, and we, by looking at BioNano, see no translocation between X and 9, but in fact we do see a translocation between X and 3, which was not reported by the karyotyping. And if we look a little bit more closely at that, where we're just looking now at the X chromosome and chromosome 3, we see there are multiple events that are going on this chromosome, an intrachromosomal translocation of 3 to 3, and at 2 different sites, translocations between X and 3. And if we reconstruct exactly what was going on the basis of this information, we find that there were 2 different variants. One is this translocation that yields a deleted chromosome of chromosome 3, like here. But the other one, which is much more unusual, was a translocation that picked up a part from X, that picked up a part of chromosome 3 in the inverted orientation, and then moved back to chromosome X to complete the area. So this new X chromosome derivative has a piece of chromosome 3 in its inverted orientation, which when we look at the banding pattern, looks identical at the karyotyping level to the tip of chromosome 9, which accounts for the misidentification of an X9 translocation. So in addition to being able to show that we can identify by the BioNano technology all the structural variants that are seen by karyotyping, we also used the information to see if we found new genes that were recurrently mutated by structural variation. And in fact, besides finding genes multiply affected in multiple cases that were both previously known leukemia driver or general cancer genes, we also found novel genes that had not been associated either with leukemia or with cancer, including ones AFAP, ENPP2, and zinc finger protein, which when we go back to the TCGA database on AML, find that expression levels of these three genes do have indeed a prognostic value, suggesting that these novel genes that we're finding as being affected by structural variation may in fact have some prognostic value. In addition, looking at the position of the structural variants relative to genes, we find that some of the intergenic region is quite important. So most of the sequencing data that is associated with clinical samples have focused on the exomic sequences, but we find that the intergenic sequences may also provide some intriguing and potentially valuable information. So this is looking at the position of translocations, inversions, gains, or deletion endpoints relative to the end of a gene. And what we find is that the endpoints of these structural variations, particularly for deletions and insertions, are enriched in the region near genes, and particularly cancer-associated genes. And this is the region in which we find promoters and enhancers of a particular gene. So we think that this, from this statistically enrichment, this may have impacts on the expression of the gene. So what might such structural variants do? Well, they could delete an enhancer and reduce expression of the associated gene, or they could duplicate an enhancer to increase the expression of that gene. Or we could fuse these structural variants could fuse topologically associated domains to collect a gene to a novel enhancer. And to assess whether or not that's the case, we looked at several cases in which a translocation was near a known cancer gene in our samples and asked whether those associated genes showed a bias in their expression. And by that I mean an allelic bias, so that this fusion affects one of the alleles of BCAL1 in this patient sample, whereas the other allele should be normal. So if we look at the expression of one allele of this gene relative to the other, which we can do by identifying from sequence analysis polymorphisms within the gene, we can identify whether RNA for this gene comes from allele A at the bottom here, carrying the C variant, or allele B above, showing the T allele. So relative to the unaffected gene, we can ask whether the translocation either increases over here or decreases the expression of its associated gene in cis by simply counting the number of genes that have the T variant versus those that have the C variant. And so this is done for this case right here for the BCL2 gene. And we can see walking across the gene, these are polymorphisms that we can identify by sequence in this gene going from left to right. As you can see, these are the polymorphisms are approximately 50%. Whereas now, if we look at the representation of those polymorphisms in the RNA, we've seen there's a strong bias where the orange variant is present much more extensively at much greater level than the blue variant, suggesting that this translocation has affected the gene, the expression of the gene in cis. We don't know whether this translocation is associated with the T or the C variant, so I can't tell you whether this translocation increased the expression or decreased the expression, but I can certainly say that the translocation has affected expression in cis of this gene. And the same thing is true for in this patient sample, in which the translocation sat next to the BRCA1 gene, and doing the same sort of analysis, we find that there's a bias, cis-acting bias in the expression of the BRCA1 gene. So in conclusion, for this half of the talk, the optical mapping identifies structural variants, all the structural variants identified by standard clinical karyotyping, but also identifies structural variants not detectable by current genomic methods. It also resolves ambiguous structural variants and detects many more related and relevant SVs. And I should point out that this technology is relatively inexpensive. It's a lot cheaper than doing whole genome sequencing. It can be done from beginning to end in 2 days. And requires no expertise other than the standard molecular biology techniques. We also note from our study that these recurrent gene disruptions may provide novel prognostic markers, and the expression levels of many AML genes are disrupted by structural variants that likely affect cis regulatory regions and would not necessarily be picked up by genomic sequencing that focused on exomes. So let me turn in the second half of this talk to the study, a recent study of HPV positive head and neck cancers. So head and neck cancers, particularly oropharyngeal squamous cell carcinomas, are one of the few cancers that are actually on the increase in this country and worldwide relative to all other cancers that are showing a decrease over the last 20 years. And this is associated with an increase in the number of oropharyngeal cancers that are associated with HPV infections, either HPV 16, 18, or 33. In 1984, only 15% or so of OPSCCs were HPV associated, where only 20 years later, that had risen to about 75%. And that number continues to increase. So we've been interested in understanding the nature of the association of HPV in head and neck cancer. And early efforts to describe what HPV doing is in oropharyngeal and head and neck cancers. It's based on information derived from cervical cancer, but the story doesn't seem to be quite the same. Now we know that HPV genome can exist in at least 2 states, either integrated in the genome or sitting episomal in the cell, residing outside of the normal chromosomes. And in fact, based on what we know about cervical cancer, the likelihood is that HPV first infects a cell, persists as an episomal element, and then at some stages integrates. And on the basis of studies of cervical cancer, the assumption is when that integration occurs, there's a loss of the E2 viral gene, which increases expression of the viral encoded oncogenes E6 and E7 which respectively um, eliminate the function of RB, promoting a rapid cellular growth, and eliminate the function of P53, enhancing structural genomic instability. So we've been interested in identifying what is the nature of HPV in these OPSCC patient samples. And we did this with a combination of, this case, optical mapping, as I talked to you before, and whole genome sequencing. And we developed pipelines to merge those data, remove polymorphisms by comparison to the blood samples from the same patients, and identify those somatic mutations and structural variants. So the first case we looked at is a 67-year-old male with a mass that you can readily see here at the base of the tongue. From biopsy, it was P16 positive, suggesting that it was HPV positive. And from the sequence, whole genome sequencing, we were able to identify hybrid reads that were partially HPV and partially human genome sequencing, and mapping the human genome sequencings that were adjacent, contiguous to the HPV, those mapped to chromosome 15. And then when we looked by optical genomic mapping at that region of the genome, we could see an insertion and duplication at that region. I want to bring your attention to the fact that in addition to this insertion and duplication at the site of HPV integration, there are also structural variations, including several translocation inversions and deletions throughout the genome of this patient sample. If we look in more detail at the structure of the region around the HPV genome, we first identified that this is actually not HPV 16, but HPV 33, and that the region surrounding the HPV 33 insertion showed a direct tandem duplication. So our assumption is that HPV 33, a fragment integrated into the genome and then mobilized the region surrounding it, approximately 20 kb, and led to a tandem duplication. So the second patient was a 61-year-old male with, again, a tumor at the base of the neck, which can be readily seen in this PET scan. Again, the biopsy indicated that it was P16 positive, so likely HPV positive. And we looked at the genome by whole genome sequencing. We saw HPV genome, but we saw absolutely no hybrid HPV genome, human genome sequencing reads. In addition, when we looked at the whole genome by optical mapping, as you can see, there are absolutely no structural variations seen either in the primary tumor or in the lymph node metastases. So we went on to look at a dozen samples, and these fell into two categories. 8 of them showed integration, and these are the viral genomes that were seen in those integrated samples. And 4 of them remained completely episomal. And as might be expected, in all cases, the E6 and E7 oncogenes are intact in these human genomes and these viral genomes, with often E6, E2, and E1 interrupted. And that's true whether or not the virus remains episomal or becomes integrated, suggesting that E6, E7 ratios relative to E2 is probably not a good metric to determine whether or not the virus is integrated. The second thing to take home from this is that there's no real structural variation or polymorphisms, as we can see by the relatedness profiles between those viral genomes that integrate, which is these two here, this one here, versus those that remain episomal. In addition, as we saw in the first case of the HPV integrating in the genome, the local area around the site of integration shows substantial rearrangements, including the tandem duplication that I showed before of the region surrounding the viral integration shown in yellow here. And that's seen in variations on that theme in multiple cases, where in this case, 90 kb of human genome surrounding the viral integration is triplicated in a tandem array. In this case, we have the virus integrating at the joint, at the junction between a translocation between chromosome 11 and chromosome 8. Another case, we have mobilization of not only the region immediately surrounding, but adjacent regions to give a complex focal rearrangement. And in other cases, we see integration at two different sites in the genome, and at each site, multiple events that are occurring, including a region in which we see tandem duplication or a complex duplication of the region surrounding this local translocation, leading to 26 tandem copies of the virus insertion, plus some of the surrounding sequences. But the really striking result that we find with this is that in every case in which the virus remains episomal, the whole genome is rock solid. It looks like very little perturbation has occurred in those genomes. Where in all the cases in which the virus is integrated, we see numerous translocations, insertions, deletions. So when the virus remains episomal, there are very little perturbations of the genome. Where it integrates, the genomes go to hell, essentially. And this is true not only at the site of virus integration, but throughout the genome at sites far distant from the site of integration. And this is just quantifying those. So here are the number of different insertions, deletions, inversions, duplications, and translocations tallied up here. These are on the left are all the ones that have integrated HPV. On the right are the ones that have episomal HPV. So highly statistically significant reduction, difference in the number of structural variants seen following integration relative to those in which HPV remains episomal. So that raises the question of whether or not does the virus integration induce genome instability, such as a consequence of increased expression of E7 that might reduce P53, or does genome instability precede viral integration? Without going into detail, our data suggest the latter. That is, that we think that HPV, as is the case with cervical cancer, first enters these oral epithelial cells and remain and set up persistence as an episomal element, and then some sort of genome catastrophe occurs, generating double-strand breaks that result in numerous structural variants within the genome, but at the same time provides an opportunity for broken ends to capture the resident HPV, leading to its integration. The second question that this poses is whether integration makes a difference. This has been a question that people have addressed for a number of years, but on the basis of our analysis, all the assays that were previously used to determine whether HPV was integrated or episomal are flawed. And so we really don't have a good measure of whether HPV in the samples that have been examined to date were episomal or integrated. But at least in our cases, it appears that those tumors with HPV integrated are more aggressive. These have a higher tumor rating, tumor scoring than those with episomal. Their mean size is larger than those that is episomal. So we validated a new technique to use optical mapping to reconstruct the genomes of solid tumors. We're able to see complicated genomic alterations that were previously unidentifiable using whole genome sequencing alone, particularly those which we see focal amplification induced by viral integration. It's a powerful tool for identifying and characterizing viral genomes in tumor. And the important point takeaway is that we're finding that an OPSCC HPV integration is strictly associated with significant genome instability. So with that, I'd like to thank my collaborators, a number of folks at BioNano, a number of folks in the Penn State Institute for Personalized Medicine, the Department of Otolaryngology and the Department of Oncology, and our multi-site clinical study on the utility of optical mapping and identifying structural variation in AML samples was done as a collaboration with Columbia University Mayo Clinic, MD Anderson, and Augusta University. With that, I'm happy to take any questions.
Excellent, Dr. Broach. Thank you so much for that riveting talk. I think we do have a few questions for you. I'm going to go in order. The first one is, can this technique be used on RNA versus DNA?
That's a good question. And we have not done it. And it would be interesting to see whether or not the methodology and the microfluidics would unwind the structure of the very often intra molecular bonding that's seen with RNA. I can imagine applying some technologies where you denature the RNA before you run it on this that might work, and it'd be intriguing to do it. One limitation right now is that the enzymology that we use to label the particular 6-base sequence on the DNA is not applicable to RNA. So we'd have to come up with a different labeling paradigm, but it'd be an interesting direction to progress.
Great, awesome, it makes a lot of sense. So the next question is, how sensitive is this method in picking up low frequency mosaicisms?
Right, that's a good question, and one that we are probing. Right now, we do the standard coverage that we get for optical mapping is 300x. So we're seeing 300, on average, 300 molecules over each region of the genome. To get a good map for which we have confidence requires 5 molecules or so. So the detection level at 300x is about 5% allelic variation. So if allele is present at a 5% level, we can pick it up. So right now, that's comparable to what's seen in karyotyping, looking at one unusual variant in one of the 20 images. The methodology has been rapidly improving, and the folks at BioNano now recommend running this with coverage at 1,000 to 1,500x, which gives coverage of mosaicism down to 1%. We can see as well as karyotyping now and anticipate that in the very near future, the sensitivity will be even greater.
Great. Another question. So very fascinating technology. and it just popped. Sorry. So how long is the sequence reads? What is the average depth of coverage? And since it has to be high molecular DNA, I presume it doesn't work on paraffin fixed tissue. So that's sort of 3 or 4 questions in one.
Yeah, no, the sensitivity is going back to the previous question. Right now we see it because we can run this at 300x. We can see at 5% allelic presence. And you're correct that the methodology requires fresh or frozen tissue or samples. It will not work on paraffin embedded fixed or formaldehyde fixed DNA samples.
Right. So 2 more questions popped up. One is a follow-up to the low frequency mosaicisms question. So the thought process there is that it may not be ready for minimal residual disease monitoring in AML, for instance, or is it too early to say?
So for minimal residual disease, I think that's correct, that we wouldn't be able to see down, even at the higher coverage, down to below 1%, which is probably the equivalent of what you would see by whole genome sequencing or by flow techniques. The one approach that we're taking is using bionano on the initial sample to identify structural variants that we can then use to design PCR-based primers that would allow us to have a sensitivity of minimal residual disease, probably down to 10 to the minus 4. So it's a combination then of front-ending with the bionano, following up with PCR-based techniques. Whether or not knowing minimal residual disease down below a level of 1%, I think is still an open question as to whether or not it will be informative in the clinic.
For sure. Awesome. Last question. So what is the reason for the increase in incidence of HPV-related oropharyngeal cancer from approximately, I guess it was about 20% in 1984 to approximately 80% in 2004? And then a follow-up on that is, would you anticipate that the HPV vaccine, given that it's come into effect after 2004, would have a change in the prevalence of HPV-related OPV?
Right. And the latter question is right on target. I think that we hope that this will be a transient epidemic of HPV infections that will be attenuated by the broad application of HPV vaccine. I think there was a mistake early on in focusing primarily on females for the vaccine. I think that that's been recognized, and more recently, the vaccine is prescribed for both males and females, and I think that will help. And it is a good question why there's been such an increase. And the simple answer that there's a change in sexual practices of young adults is probably unlikely. I don't think that people do things differently now than they did 100 years ago. And that suggests that perhaps there is a change in the nature of the virus that is floating around that may actually increase its infectivity or virulence. But that's an intriguing epidemiological question. And I do scratch my head every now and then wondering why there's been such an increase in incidence.
Great. Well, thank you so much for all those questions.